So why don't we get started? So it's my pleasure to introduce Marie Guerraty, who's visiting us from UPenn. Marie did her undergraduate at Duke and majored in mathematics and minored in biology, and then went to UPenn where she got her MD and PhD. And then she stayed on at UPenn for residency and fellowship, and now is an instructor and has been focused in on studying microvascular disease at multiple levels. So studying at a very fundamental level, and then also studying it in translational small animal models and actually even in patients using dynamic PET imaging. So join me in welcoming Marie.
Good morning. Can you hear me well? Yep. Thanks so much for the invitation to be here so that I can share some of this work with you. I'm going to be telling you a little bit about coronary microvascular disease and how I think it's next frontier in ischemic heart disease. So after 40 years of progress in decreasing cardiovascular mortality, these efforts and the progress has stalled recently. You can see the curve, which was coming down nicely, hasn't actually just stalled, it's rebounded up. And there are many reasons why this has happened. One is healthcare disparities. We're not able to get the right treatments to the right patients at the right time. There are also rising rates of obesity and diabetes that contribute to this. But there's also a contribution from coronary microvascular disease and the fact that we've been underappreciating a significant portion of the coronary microvasculature. Because of these curves, it's pretty clear that we need new out-of-the-box ways of approaching ischemic heart disease, new therapies, new imaging modalities, new diagnostic modalities. So the coronary microvasculature is structurally and functionally pretty complex. It's composed of arteries and veins and lymphatics, but mostly arteries of different sizes that have pretty specialized functions. The larger conduit vessels that are greater than 500 microns in diameter are the named coronary arteries, the RCA, the left circ, that we image with coronary angiograms. These vessels develop coronary atherosclerosis, but functionally they don't do all that much. The microvessels, on the other hand, is where all the physiology happens. This is where all the action is happening. So these are the vessels that are a little bit smaller, so 200, 100 microns, and actually all the way down to the capillary level. These are the blood vessels that regulate blood flow to the cardiomyocytes. These are the blood vessels that vasodilate in response to flow, and these are the blood vessels that are sensing the local metabolic environment and regulating blood flow to meet that need. So for 40, 50 years, we've been studying coronary artery disease, which affects these larger conduit vessels, and we've been studying coronary atherosclerosis, the cell types involved in coronary atherosclerosis, and treating coronary atherosclerosis. And I think the next phase of treating ischemic heart disease is going to be focused on the coronary microvasculature, these smaller vessels. We know a little bit about the pathophysiology of coronary microvascular disease. We know that there are some structural changes. There's luminal obstruction or vascular wall thickening. You can get immune cell infiltration of the vessels. We know that you can get dysfunction of the vessels and endothelial dysfunction. And we've all seen what this can look like clinically. So patients present with MIs or with angina. They have some sort of objective evidence of ischemia, either on a stress test or they have leaked troponins. They get a coronary angiogram which shows that there's actually no obstructive coronary artery disease, and then clinicians put the pieces together and say that this must be microvascular disease. So one way of thinking about coronary microvascular disease is ischemia in the absence of obstructive coronary artery disease. There's a lot of clinical evidence that this is a pretty important disease process. So there's a recent growing interest from the Women's Ischemia Syndrome Evaluation Study that was summarized in the 2017 white paper. And this talks about ischemia with no obstructive coronary artery disease as INOCA. This paper that came out of Europe looked at 12,000 patients that had convincing exertional angina symptoms. They were convincing enough for a cardiologist to feel that these patients needed a cath. And when they got their cath, 60% of women and 30% of men actually had no obstructive coronary artery disease. Yet these patients had worsened outcomes compared to control patients. So they don't have obstructive CAD, but they have something that increases their cardiovascular risk. And then more recently, this is the verdict trial that came out in 2018. The entire purpose of this randomized controlled clinical trial was to enroll patients that were having an NSTEMI and randomize them to cath and revascularization at 24 or at 48 hours. There was no difference in the timing, but what I found really interesting is that 1/3 of these patients actually didn't have any obstructive coronary artery disease. So we're seeing these patients pop up in a lot of these big trials that we've been doing. And I think CMVD is an important clinical entity to address now for the following reasons. So it contributes directly to cardiac morbidity and mortality. Patients have symptoms from this, they present to the emergency room with this, and they can die from this. CMVD also worsens outcomes for patients that have coronary artery disease or patients that have an MI. The biology of coronary microvascular disease is very closely related to collateral formation, which we know to be predictive in ischemic heart disease. There's also recent growing evidence that coronary microvascular disease and pathology may be part of the pathogenesis for heart failure with preserved ejection fraction. And for HFpEF at this point, we are still just trying to understand how it's developing to try to come up with targeted therapies that might perhaps be upstream of the clinical syndrome that we're seeing. And we also know that microvascular function plays a really important role in various cardiomyopathies like hypertrophic cardiomyopathy. Technological advances in cardiac imaging are now making this field accessible to clinicians. So we're finally able to measure surrogate markers of the coronary microvasculature with cardiac PET and MRI and some echo modalities. But yet we still have limited evidence-based treatments and actually no targeted therapies. So in a perfect world, the next step would be to do an unbiased assessment of coronary microvascular disease and come up with some targets. So we could think about doing a genome-wide association study for CMVD to enroll patients that have CMVD and patients that don't have CMVD, genotype them or sequence their genomes, and look to see if we can find some genes that might be related to CMVD. But we don't live in a perfect world. We don't have any coronary microvascular disease GWAS studies, and it's actually pretty hard to design these studies because it's hard to phenotype these patients. The phenotype is a lot of different data that is brought together by the clinician. But what we do have are thousands and thousands and thousands and hundreds of thousands of patients that have been enrolled in coronary artery disease GWAS studies. These are actually really ischemic heart disease studies. For example, if you look at the Procardis enrollment criteria, patients got enrolled in this coronary artery disease trial if they had one of two events. They could have an MI or symptomatic acute coronary syndrome. So to have an MI, I put in bold criteria that you could meet if you only had microvascular disease. You had to come in with typical ischemic chest pain and leak some troponin. You didn't necessarily have to have coronary artery disease. Similarly, to be classified as having symptomatic acute coronary syndrome, you could present with an unstable angina type picture and have chest pain at rest with dynamic ECG changes, and that would get you enrolled. So there are clearly some coronary microvascular disease patients that are clumped into these ischemic heart disease GWAS studies. Additionally, I've been really interested in the fact that developmental programs are often recapitulated in disease states. And we think about this a lot in heart failure when fetal programs are turned on and there's a switch in metabolism back to glucose. So we think about this a little bit in cardiology, but it's always fascinated me that there's a lot of cardiac development that we are not drawing from to get clues about what's going on in disease states. So my approach to CMVD is based on those two things. One, that coronary artery disease GWAS loci are actually ischemic heart disease loci, and contained in those 200 and some genes that we have right now, or 200 and some loci, we actually have a lot of information about coronary microvascular disease. And there are genes that are involved in coronary development that we're learning about that might be really relevant in coronary microvascular disease. So based on this framework, I focused on two molecules that I'll tell you a little bit about. One is FOG2, which is a cardiomyocyte transcriptional coregulator. And the other one, TWIST1, is a transcription factor that is active in endothelial cells, but also in smooth muscle cells. So FOG2, or Friend of GATA 2, is a really, really neat molecule. If you knock it out in mouse embryos, they don't develop coronary microvasculature, so it's crucial to develop coronaries. But if you look at the role of FOG2 in adults, if you conditionally delete, so if you turn off FOG2 in an adult mouse, within weeks, the microvasculature of that mouse recedes. So you get capillary rarefaction, and the whole vascular tree, instead of being nice and plump and covering the whole heart like you see up here, becomes very, very thin. And of course, this results in heart failure in the mouse. The mechanism of this is that FOG2 is regulating an angiogenic program. So FOG2 is increasing expression of proangiogenic factors like VEGF and FGF2 and 9, but FOG2 is also decreasing angiostatic factors like the TIMPs and different collagen factors. So FOG2 in mouse development and FOG2 in adult mouse coronary microvascular homeostasis plays a really important role in regulating these programs. So this led us to the hypothesis that FOG2 might play a role in adult human coronary microvascular disease. So in order to set out to see if this is true, we started doing some PheWAS analysis. So you're all familiar with GWAS analyses, where we take a single phenotype, like coronary artery disease or serum LDL levels, and we check the entire genome, color-coded by chromosome, to see if there's an association between any locus and the phenotype of interest. PheWAS is a converse of that. It's a Phenome-Wide association study. So we have a genetic variant or a group of genetic variants, and we look at all ICD-9 and 10 diagnoses codes, color-coded by organ system, to see if that variant might be associated with some sort of phenotype by ICD-9 diagnosis. And in order to do this, we used the Penn Medicine Biobank, which is a collection of patients that have been enrolled at Penn. For this particular study, we had about 12,000 patients, and these are patients that have interacted with the healthcare system and consented to be enrolled. So they're not clinical trial patients, they're not particularly well phenotyped. We may or may not have cardiac testing on them, they may or may not be seen once in the emergency room, or maybe they've been followed by a cardiologist every 6 months for years. So we kind of get what we get with those 12,000 patients, but we do have whole exome sequencing information on those 12,000 patients. So to ask the question if FOG2 might be relevant to adult human coronary microvascular disease, we looked at 34 non-synonymous variants in the FOG2 gene, in the coding region of the FOG2 gene, and the 3' UTR. And of those 34 variants, we actually found one that in PheWAS analysis was associated with a human phenotype. And that phenotype was an ICD-9 diagnosis of atherosclerosis, which is a great diagnosis because it's not really something you give someone when they have chest pain or when they're presenting to an emergency room. It's kind of an imaging finding. So this variant, which is RS28374544, is actually an A to G substitution or mutation or variation, and this results in an amino acid switch from a serine to a glycine. So here's a picture of the FOG2 protein. It's actually pretty big, and this is where that variant manifests. And what's interesting is that FOG2 has been studied in the context of mouse development and also congenital heart disease. There are different FOG2 mutations that have been associated with complex congenital heart disease, and this is one of them actually. So when we stumbled upon this finding, we went back and looked and said, hey, people have been talking about this mutation because they think it's associated with congenital heart disease, specifically TGA and tetralogy. But what was really interesting is what this variant looked like in our population. So first of all, it was really well represented. We had 44 patients that were homozygous for the minor allele, that had a GG genotype. We had 550 heterozygous patients, and we had almost 11,000 AA or control or wild type patients. This represented a minor allele frequency of 0.03, which is actually in line with the minor allele frequency of this variant, not just in the Penn Medicine Biobank, but in some of these other databases like the EXACT database. What's really neat is if you look at more homogeneous populations, like populations of African patients, you see that this variant is much more highly represented. And if you look at populations that are Caucasian or Asian, this variant is not really present at all. So this variant is actually pretty African-American specific in our population. The other thing we found that was pretty interesting is that in our adult population, this variant was indeed associated with complex congenital heart disease. So of our 44 homozygous patients, we had 2 complex congenital heart disease patients, 1 tetralogy and 1 TGA that had been repaired when they were born. We also saw 1 in our heterozygous population. But what's really interesting is 42 of our 44 patients did not have congenital heart disease. So what did those patients look like? So when we looked at discrete left heart catheterization hemos for these patients, we saw no difference in systolic aortic blood pressure during invasive left heart cath. We saw no difference in LV-EDP or wedge pressures or RA pressures. We saw no difference in heart rate for these patients. When we looked at echo parameters for those patients on whom we had echoes, we saw that the EF was the same in all these patients. Stroke volume, left atrial volume, and diameters and wall thicknesses were all the same. So we weren't seeing a lot of differences to explain why diagnosis of atherosclerosis might have gone up, but we also were not focusing on vascular phenotypes for these patients. When we looked at what these patients had, they were actually presenting to the emergency room with chest pain. So patients with the GG genotype actually presented much more frequently than the other genotypes. And if you look at the survival curve, these patients actually had different survivals. So we didn't see any difference in actual MIs. We didn't see any difference in outpatient angina presentations or heart failure, but we did see a difference in patients coming in to the emergency room with chest pain. The problem with that set of data is that it's not matched. So the 44 GG patients are actually mostly African American, they're 60% female, and they vary in a risk profile, in the risk factor profile. So to do deeper phenotyping, we had to generate some matched cohorts. So we took our 44 GG participants, and then we found 88 heterozygous and 88 AA participants that were matched based on age, gender, and race. And as you can see, they're 93% African American. The cardiovascular risk factor profile for these groups was actually pretty similar. We did see a difference in a history of the diagnosis of atherosclerosis here, which is reassuring because that was our initial GWAS signal to begin with. So we looked at coronary artery disease in these different cohorts. We manually abstracted coronary angiograms by a blinded reviewer and just counted the number of obstructed vessels that they had. And what we saw was that patients with the GG genotype had fewer obstructed vessels, so they were less likely to have 3-vessel disease, more likely to have no vessel disease. We then looked at whether these patients were meeting clinical criteria for CMVD. So a little bit like that slide I showed you earlier, patients had to have symptoms of angina or MIs or exertional dyspnea. They had to have symptoms that were not due to another cause. So if a patient had a restrictive cardiomyopathy, didn't really matter that they were dyspneic, it was not attributed to CMVD. We considered whether or not they had a positive stress test, but this was not a requirement to count them as having CMVD. But they had to have a cath, and that cath had to have relatively clean coronary arteries. And when we used these pretty strict criteria for our participants, again, for many of them, we didn't have enough data to include them in this count. But what we found is 3 of our 44 participants that were GG actually met those criteria, whereas only 2 of the heterozygous and none of the AA participants met those criteria. So to get yet another measure of microvascular flow or function, we turned to their coronary angiograms. Let's see if I can get these to play. So when you inject contrast dye into the coronaries, it can flow quickly or it can flow slowly. And if there is not an obstruction proximally, the coronary microvasculature and the resistance vessels are determining the rate at which that contrast dye flows. And in some patients, the flow is much, much slower than in other patients. Here's a pretty normal one, and then in this one, there's a little bit of low flow. If this is really extreme, then we call it a low flow phenomenon, and there have been cohorts published looking at patients whose flow on coronary angiogram is really low. But there's actually a pretty dynamic, there's a gradient. So some patients are totally normal, Some have low flow where you can look at them and you see that the dye is barely inching its way along. And this one, it's noticeable but not terribly severe. So the TIMI frame count is a way to quantify how quickly the dye is moving down the vessel. And what we found is when we counted the number of frames it takes the dye to move from a proximal landmark to an established distal landmark as a semi-quantitative measure of microvascular flow, the GG participants had altered myocardial blood flow by this criteria. So reassured that there might be something going on with these patients, that FOG2 may actually be affecting the coronary microvasculature, we dug a little bit deeper. So this A to G mutation at the 1968 base pair position results in a serine to glycine switch. So when we did some peptide modeling of this small region, which again is right here, we found that if you have a polar serine at that residue, then you maintain this loop structure. But if you switch that polar serine to a glycine, which is the smallest amino acid, and it can introduce a lot of mobility into a peptide, suddenly you lose that secondary structure. So we then decided to look at FOG2 wild type and the FOG2 variant or mutant to see what it might be doing in cells. So for a lot of these experiments, we use the cell line AC16 cardiomyocytes. It's actually a human cell line, which is great because it has human genome in there. It also is a cell line that grows really nicely like HeLa cells. It is pre-contractile. So if you're studying sarcomeric biology, it's not very useful. But if you're studying transcriptional programs in cardiomyocytes, it's actually a great cell line. And we put into these cells FOG2 or mutant FOG2 to see what FOG2 might be doing. And it actually wasn't doing too much. So FOG2 is a transcriptional co-regulator. So even if you increase the amount of FOG2 or mutant FOG2 in a cell 1,000-fold, it actually doesn't do too much. We didn't see a significant difference in most of the known targets of FOG2. But since FOG2 is a transcriptional co-regulator, it needs a partner, it needs a binding partner to actually have transcriptional activity. So this made us think that we should find a good binding partner for FOG2. GATA4 is known to be a binding partner for FOG2, but we hypothesized that FOG2 may also interact with HIF. So FOG2 is expressed at the same time frame in development as HIF, and they actually share a really important target in VEGF. So I'm sure most of you have heard about HIF by now. The Nobel Prize was just awarded to Francis Crick, Peter Radcliffe, and Bob Kalin for their discoveries in HIF. And it's because HIF is such an amazing cellular pathway. So in the presence of hypoxia, the HIF protein is stabilized, and you get a buildup of the HIF protein. The HIF protein can then go to the nucleus, and it leads to transcription of a bunch of genes. You can classify those genes two ways, angiogenesis and mitochondrial function genes. And this actually makes great sense if you're a cell. If you're a cell and you're not getting enough oxygen, you want to do these two things. You want to increase angiogenesis, get more blood vessels to bring you more oxygen, but you also want to decrease your reliance on oxidative phosphorylation and decrease your utilization of oxygen. You want to switch away from oxidative phosphorylation. So HIF regulates both of these. It promotes angiogenesis, but it also dampens down mitochondrial function. And if you're a cardiologist or you have a cardiac-centric view of the world, one of these is really good, and one of these is really bad. And when they've manipulated HIF in mice, actually the result is that bad things happen. So if you overexpress HIF, sure you get more angiogenesis, but you end up getting heart failure because you dampen down mitochondrial function. Similarly, if you don't have enough HIF, you don't get appropriate angiogenesis. So manipulating HIF at that level is not a viable option for the heart. So what HIF does in these AC16 cells is the same thing that it does in pretty much every cell. So if you treat the cells with hypoxia, over a matter of hours, the HIF protein builds up, it goes to the nucleus, and you get transcription of these targets. VEGF-A is an angiogenic target, and PDK1 and BNP3 are metabolic targets. So the transcription factor just goes and does its thing. We wanted to ask the question, does FOG2 affect how HIF does this job in cardiomyocytes? So to do that, we used CRISPR-Cas9 genome editing to make cells deficient in FOG2. So AC16 cardiomyocytes, like all cardiomyocytes, are diploid or more, so it's actually pretty hard to knock down all the alleles, but we were able to knock down 1, 2, or 3 of the alleles. So these are knockdown, not knockout cell lines. And you can see from, here's an example of a control. There's some FOG2 expression, and in a lot of these knockdown cell lines, the FOG2 RNA expression is decreased. Similarly, FOG2 protein is expressed at low basal levels in the control, and in the knockdown cell lines, FOG2 is decreased. This doesn't really affect how much HIF the cell has. And we saw something really interesting. So when we took these knockdown cell lines and we treated them with hypoxia, we saw no difference in how the cells responded to these metabolic genes. So expression of PDK1 went up over hours, BNIP went up over hours, and COX4-1, which is another metabolic factor, went up, stayed the same over several hours. Angiogenic factors, on the other hand, did something pretty neat. So VEGF usually increases in response to increased hypoxia and HIF, but in the FOG2 knockdown cell lines, we saw that this response was pretty blunted. Similarly, we looked at adrenomedullin and saw the same response, and we also looked at some of the ANGPT1 and 2 genes and saw something similar. So in other words, it looks like modulating FOG2 actually affected how HIF induced its angiogenic and metabolic target genes. So instead of just having this model where hypoxia leads to increased HIF, and we get both of these arms, it seems that FOG2 may help HIF target some of its target genes and not all of them. So there are clearly a lot of really interesting therapeutic potentials that may come from this. But if we go back now to our FOG2 and our FOG2 mutant and say, okay, well, when we overexpress FOG2 wild type and mutant, we didn't see too much. Well, what if we do that in the context of hypoxia? And then we started seeing some interesting differences. So if you transfect the cells with an empty vector versus the FOG2 versus the FOG2 mutant, you start to see that there's a pretty big difference in how these cells upregulate angiogenic genes in response to hypoxia. To further characterize this, we didn't just look at the gene expression in the cells, we took conditioned media from the cells that had been treated. So the cells are making more VEGF and ANGPT1, but what are they secreting into the media? What signals might be getting out? And we found a difference in some of the ANGPTs and some other inflammatory markers. So we can take that soup that's different between FOG2 and FOG2 mutant and put it on endothelial cells to see, all right, if a cardiomyocyte is expressing FOG2, or if it's expressing the mutant form of FOG2, does that affect how it signals the neighboring vascular cells to make or support vessels? And in a tube formation angiogenesis assay, we saw that they're actually, this is what they look like, but we saw that the FOG2 and the FOG2 mutant behave differently. So cells that had been treated with FOG2 or the FOG2 mutant formed different amount of branches, of segments, the length of the branches were different. So we started seeing some subtle angiogenic difference between these. So to summarize this part, we saw that in humans, this FOG2 variant results in a CMVD-like phenotype. FOG2 may serve as a cofactor for HIF and modulate how HIF induces its angiogenic targets and how it may do that differently from its metabolic targets. We saw that the mutant alters a HIF-induced angiogenic program in vitro, but we haven't yet determined what the effect of this mutation might be in terms of coronary microvascular physiology, which is what we need to do to tie it back to the patients. So moving forward, we have to take the wild type and the mutant and start to evaluate them in the context of a model system so we can actually see what these different angiogenic factors mean for the vasculature. In order to evaluate that model system, we have to have a way to quantitatively assess the coronary microvasculature in mice or your model system of choice. So the direct goal of this is to find a non-invasive method to quantify microvascular function and then use this to phenotype mice that have that A1968 gene mutation. There are lots of cardiac imaging options for mice, including MRI, PET, and echo. We have used coronary Doppler of the mouse left coronary to get coronary flow reserve. It's really technically challenging. And we're also working to develop mouse SPECT. So this is some mouse SPECT imaging that we've done using a pinhole collimator, and this is very similar to what we do clinically with cardiac PET. So what you can do is take pretty short frames, so these are 10-second frames, and image the mouse heart as technetium-99m sestimide is injected venously. So you can see what the curves do here. The blue curve is the blood pool, which is this little red box in there. So initially there's no activity, and then as the tracer reaches the left ventricle cavity, you see a huge burst in activity, and then over time, as the tracer gets taken up by the myocardium and other tissues, the activity in the blood pool goes down. And similarly, in the myocardium, which is this blue region of interest, you can see that there's no uptake, and then as you get the bolus of the tracer, you get steady uptake into the myocardium. You can model this to actually get a K1 value and try to link that back to myocardial blood flow. And this is what we do with our patients in cardiac PET. But it's really tricky in mice. So some people, including groups here, have described using intramyocardial blood volume as an alternate measure of the coronary microvasculature. And this is based on the idea that there's a relationship between flow and volume, actually, and a lot of the volume, the intramyocardial blood volume, is actually contained in the coronary microcirculation. So to leverage this in mice, what we did is we labeled red blood cells with technetium-99 pyrophosphate, and we can image the same regions of interest, the blood pool and the LV, and track activity over time. And we can give the mouse a hyperemic insult, whether it's more isoflurane to vasodilate or dipyridamole to vasodilate. What you can see is if the blood pool is kept constant, the amount of uptake in the left ventricle goes up. And this means that the mice are able to increase under hyperemic conditions their microvascular blood volume about 22%, which is similar to values that have been found in dogs. So initially, the goal of some of the imaging work is to have a quantitative, non-invasive readout to assess the mutant mouse that I'm going to study next. But this imaging modality actually has thousands of different applications. It would be incredibly useful to study potential therapies for coronary microvascular disease, and to also evaluate other molecular gene targets that might be involved or might play a role in the pathogenesis of microvascular disease. And now I'll get to the TWIST1 story, which is an example of this. So the genome-wide association studies that have been performed for coronary artery disease have given us 200 and some loci that are associated with coronary artery disease. Lots of groups have spent a lot of effort to do functional genomic studies and figure out how this variant, identified by GWAS in people, actually affects different cell types to cause coronary artery disease. This is one locus that has been described, 210-7595, and then this is its position on the genome. So it's located here between HDAC9 and TWIST1. And it's actually, this variant has been linked to HDAC9, and there has been a lot of research in the role of HDAC9 in coronary artery disease and some other phenotypes because it's geographically closer. But we showed in our studies that this locus is actually regulating TWIST1 gene expression as well as HDAC9. So we found that participants that had the minor allele for this variant actually had more TWIST1 transcripts than the GG genotype. So we looked at whether TWIST1 may be playing a role in endothelial or smooth muscle cells, and given that the expression of TWIST1 is much greater in smooth muscle cells, and a lot of the regulatory regions are actually enhancers that are active in smooth muscle cells, we examined the role of TWIST1 in smooth muscle cells. And what we found in a proliferation assay is that if you overexpress TWIST1, which is this pink line, number of cells goes up. So TWIST1 promotes smooth muscle cell proliferation. Conversely, if you use siRNA to decrease the endogenous amounts of TWIST1, the cells really don't grow. So TWIST1 is really important for smooth muscle cells to grow and proliferate. A different smooth muscle cell phenotype is calcification. So smooth muscle cells have the ability to become osteoblast-like and form calcifications. And one way to test that is you can take smooth muscle cells in culture and give them a phosphate-rich media, and they will develop these little black calcifications that are stained by alizarin red. So when we did that, we found that if we overexpress TWIST1, we decrease the number of calcifications. Conversely, if we decrease the amount of TWIST1 by using an siRNA against TWIST1, we increase the number of calcifications. So this was a pretty interesting result that TWIST1 may play a pretty significant role in modulating smooth muscle cell phenotypes. We next reached out to a mouse model to see if we could understand what this might mean in vivo. So we took myosin heavy chain 11 Cre mice. So these are mice that have a floxed TWIST1 allele. And when you combine the Cre and the flox, you remove the TWIST1 gene only from mature smooth muscle cells. And then we take these cells, these mice that have TWIST1 deleted from their smooth muscle cells, and we give them an atherogenic stimulus. And the first thing we found is that there was actually no statistically significant difference in the amount of atherosclerosis. So the TWIST1 knockout mice only had TWIST1 deleted from a tiny subset of their smooth muscle cells, and then this did not affect how much atherosclerosis developed over a few months. But when we looked a little bit closer, we saw something really interesting. So these are immunofluorescence sections of the ascending aorta, and these are from a wild-type mouse where you have the shoulder region of an atheroma that has developed in the artery, and then you have kind of the middle of the plaque here. And what you see is that there are a lot of smooth muscle cells in the vessel wall when we stain with SM22A, but there are also some smooth muscle cells overlying this atheroma. In the middle of the lesion, you have a lot of SM22A positive smooth muscle cells overlying the atheroma. In the knockout mouse, we didn't see that. So if you look at the shoulder region, you don't see a lot of smooth muscle cells there. There are still a lot of smooth muscle cells around the vessel wall, but not as many overlying the atheroma in the shoulder region. And when we looked mid-lesion, we also didn't see the same number of smooth muscle cells. And then when we looked at calcification, just like we had done in our in vitro studies, we saw that the wild-type mice, so this is a cross-section at the base of the aortic root, so you have a little bit of the aortic valve leaflet here, and then this is the root with a big hunk of atheroma. And if you look at the subendocardial calcifications, we don't really see any here, but you do see a lot in the knockout mouse. So the knockout mouse is forming subendocardial calcifications, whereas the wild type is not. You also see some differences here in valve calcification, where they're both forming calcifications, but the knockout mouse is clearly forming more aortic valve calcifications than the wild type mouse. When we quantified these, we saw that there was a difference. So it's not just these pictures, but when you quantify it over a large cohort, we see that. So it looks like TWIST1 may play an important role in smooth muscle cells in vivo, and that role may be to affect plaque morphology. So we found that this locus, RS2107595, is associated with increased TWIST1 levels, and then we evaluated what TWIST1 might be doing in smooth muscle cells. In our in vitro studies, we showed that TWIST1 might increase proliferation and decrease calcification. And similarly, in our in vivo model, we saw that the TWIST1 knockout had fewer smooth muscle cells overlying lesions and had more calcification. So the way we put this together is that TWIST1 may actually modulate the developmental stage or the phenotype of smooth muscle cells. And smooth muscle cells are known to be really important in coronary disease, and they're able to modulate their phenotype and promote or protect against atherosclerosis based on that. So if you have a smooth muscle cell that is driven to be more proliferative, it will grow, but it will not have the potential to form calcifications. If you don't have TWIST1, you push the cells towards a more developed or a more specialized function. They become more contractile, and you can even push them to be osteoblast-like and form calcifications, but these cells don't really proliferate well. So TWIST1 is playing a role, we think, in coronary artery disease by modulating how the smooth muscle cells behave. And again, a lot of the in vivo stuff I showed you is based on deleting TWIST1 from a tiny subset of smooth muscle cells, but we haven't deleted TWIST1 from larger populations, and we then may be able to see a more dramatic phenotype. So if we want to connect this finding back to human disease, increased TWIST1 has actually been associated with stable coronary artery disease. So of the 200 loci that we have found for CAD, people have been doing subphenotyping of those, saying that coronary artery disease that includes MI and stable angina is actually a pretty heterogeneous group. So if you look at a locus, you can tell whether or not that locus is associated with a more stable CAD phenotype or an MI phenotype. And this is one paper that did that. So they found, for example, that the ABO locus is associated with MI over CAD. The TWIST1 or HDAC9 locus, on the other hand, is actually associated with stable symptoms. So it's possible that TWIST1 may contribute to plaque stability and let patients form larger plaques that therefore don't rupture and they get a stable CAD type symptom. Alternately, and this is where I think there's a lot of interesting potential, TWIST1 affects smooth muscle cell phenotype, and the smooth muscle cells are really important in regulating coronary microvascular tone. So it's possible that if we disrupt where on this spectrum the smooth muscle cells in the coronary microvasculature sit, we may lose some microvascular regulation and then end up with dysfunctional microvasculature. So another aim of developing the mouse imaging is to be able to evaluate the role of these coronary artery disease GWAS loci genes in the context of microvascular disease, starting with TWIST1. So in summary, coronary microvascular disease contributes to the burden of ischemic heart disease. It's an understudied area of coronary vascular biology, and it has pretty direct and definitely widespread clinical relevance. So my approach to coronary microvascular disease has been to use known coronary artery disease GWAS loci and genes, and also genes that are known to play a role in coronary development. We found that a variant of FOG2 is associated with human CMVD and may affect how HIF regulates an angiogenic program. And we also showed that FOG2 may be acting as a cofactor for HIF. FOG2, because of that, may be a really interesting therapeutic target, since it allows us to manipulate part of the HIF pathway without actually directing a transcription factor. And TWIST1 we show is a smooth muscle cell phenotype regulator, and this approach and general methodology could be applied to lots of genes and different targets to gain novel insight into coronary microvascular disease and potentially find new therapies. So a lot of people have helped with this project, and I'm happy to answer any questions.
Really wonderful, fantastic stuff. I was wondering, in the FOG1 story or the GWAS study, did you find other potential targets besides FOG1? You must have. Like, do you have any? Can you give us any insight into some of those other targets?
Well, one of the targets that meets both of those criteria is ENOS, and I know there's been a lot of interest in that. So that's a really good example of a coronary artery disease GWAS gene and locus that clearly plays an important role in the microvasculature. And I haven't pursued that or studied that, but I think that's another pretty low-hanging fruit for that specifically. A lot of the coronary artery disease GWAS loci also affect lipid levels and LDL, and we know that those, in some not necessarily direct way, affect the microvasculature. So I think there's a lot of interesting biology that we can link that's either known or unknown. A lot of the GWAS targets actually aren't even known. They're genes and we don't even really understand their function. So whether or not they may play a role in microvascular disease is wide open.
All right. Very nice talking to you about human genetics and your work. I have two conceptual questions. One is that about your genetics, and that is the FOG2 that you showed is the common variant in African American. You compared it with the actually general population of control, I think black and white together.
Yes.
And so I think it would be more validated if you would have actually compared the black controls than not the total.
Yes.
And then if you had done Bonferroni correction or not, I don't know what would be the p-value. But another conceptual question is that I link it to the microvascular disease. And you are actually testing two neural crest genes. TWIST1 and FOG2 are basically expressed in the neural crest and actually involved in epicardial vessels. And Specifically in your knockout in the FOG2, I don't know what type of knockout you had on. That was a targeted. I don't know in what tissue.
That was so the FOG2 conditional knockout that I showed was Bill Pugh's work, and they conditionally deleted FOG2 actually from cardiomyocytes, endothelial cells, and smooth muscle cells, and they found that it's cardiomyocyte FOG2 that is important for this phenotype. It actually doesn't do much in endothelial and smooth muscle cells.
So then you actually was knocking it out also in the endothelial muscle cells. So, but bottom line is that it seems to be more linked through actually macrovascular because a lot of your phenotypes are macrovascular. Also, the third question, I hope I'm not asking too many questions, is that the common variant you find in the second gene, was it a functional variant? I mean, meaning that GWAS finds a lot of variants, but they may not be exactly functional. They may be associated with another variant that is actually functional. Did you guys work on to find what is the functional variant?
We did some fine mapping to determine if this was indeed the lead SNP. I don't know with 100% certainty that it's the lead SNP, and it's hard to differentiate whether that's lead SNP or whether there are 4 lead SNPs in that haplotype block that are really important. We did do some studies where we did luciferase studies, so we mutated that single base pair and actually saw that it affected how that region worked as an enhancer. So you're right, I think we don't have definitive proof that it's the lead SNP, but based on in silico modeling of what it would do to that enhancer region, I think we're confident that it's probably very important.
Wonderful work. And back to the FOG2 story. So some of the effects on target protein expression or target gene encoding protein expression were pretty dramatic, and maybe even more dramatic than effects on RNA level. So I was wondering whether there was any, and that's again the problem with GWAS is looking at loci encoding regions, et cetera, whether there were any non-coding indirect effects of FOG2, not so much as a transcriptional coregulator of HIF, but affecting non-coding RNAs, microRNAs, and that could have a sort of a multiplicity effect on numerous genes.
Yeah, I think that's a really good point because there is a very long antisense transcript that gets made for that distal part where a lot of these variants would also hit. The role of that antisense transcript is, again, not really known, so it could absolutely have a role in that. Yeah. And then the other interesting thing to your point, the locus in the FOG2 gene that's been associated in coronary artery disease GWAS studies is actually intronic and far from this. So I looked at coding variants as low-hanging fruit for the PheWAS, but figuring out the genetics of why this intronic variant has been, this isn't the one that's been linked with coronary artery disease. It's an intronic variant. So figuring out what they're doing and whether they're acting through FOG2 or whether, like you're saying, they're regulating a linked RNA that then goes on and affects a different gene. And I think that's why the field of functional genomics is so challenging and frustrating.
That's true. Yeah.
First of all, welcome and great talk.
Thank you.
Great work. These are kind of a little far afield, these questions. So one is that the first question related to the fact that there have been associations clinically, maybe with regards to differences by sex as well as in patients who might have stress response differences as well, and coronary microvascular disease, and how would you explore that in these models? And the second question really relates to next steps, and obviously there's been a lot of interest in Mendelian randomization kind of projects, UK Biobank, as an example. Do you see this moving in that direction, and what are your next steps personally?
So I think to address the sex issue, I think there's a really interesting interplay between gender and microvascular disease, because traditionally, historically, cardiac syndrome X was a woman's disease. And now in some clinical studies, we've actually found that, for example, if you use cardiac PET, the rate of microvascular dysfunction by cardiac PET is the same in both genders. But what I think might be going on is that men tend to form more visible focal coronary artery disease. So for that huge segment of the clinical population that has both microvascular disease and CAD, the men are getting treated because they have something that just looks a little more prominent. So they get binned as having coronary artery disease. They get put on aspirin and statin, maybe a beta blocker, and they're treated more aggressively. And that's why we see differences in outcomes. I don't think there is necessarily a huge difference in whether men or women are having coronary microvascular dysfunction. There are some physiologic differences. So resting myocardial blood flow, for example, is higher in women, and MBFR tends to be higher in African Americans. So there are some gender and racial differences to the coronary microvasculature. And whether that is from something like a FOG2 variant that is more expressed in African Americans, that remains to be seen. But I think it's a really interesting area to investigate to see if maybe some of the disparities actually have a biological source. And then I think moving forward, the framework of looking at coronary artery disease GWAS studies and getting a subset of those that are involved in coronary microvascular disease uses things like the UK Biobank and a lot of data that's been collected and resources that exist and trying to find additional targets. So that is one area that I would definitely love to pursue. And then the FOG2 and TWIST stories, specifically figuring out how they affect microvascular function in a mouse model, and what that translates back to humans to mean, I think is another direct area that I'd love to work in.
I have a question to go back to TWIST1 and modulation of smooth muscle cell function and its potential role in the myogenic response as it relates to the microcirculation. And there is some data that integrins are involved in that myogenic response. Have you looked at changes in integrin activity?
No, I think the very easy next step for this is to just look at the mice that have these differences and do broader profiling to see, are the hearts of these mice expressing different smooth muscle cell factors like integrins? Are they expressing different PDGFs and things like that?
Aaron.
Very nice and broad, really talk. That's great. Do you think microvascular disease is an endothelial problem, smooth muscle problem, or inflammatory problem?
Or what's the third option?
Inflammation.
D. I think it's a cardiomyocyte problem. So a lot of the FOG2 work has been very cardiomyocyte-centric, and I think we know that hypertension, diabetes, poor periodontal health, all of these things, hypercholesterolemia, are vasculotoxic. So they affect endothelial cells, they affect macrophages, they affect inflammatory cells, they affect smooth muscle cells. But what I'm interested in is in understanding how the cardiomyocyte helps to regenerate against that, because I think it's very important to treat hypertension, and it's important to treat diabetes. But one step further is you already have the vascular insults. What can we learn about the cardiomyocyte that promotes collateralization, that promotes a healthy compensatory response to those vascular insults?
Follow Marian's question, and based on the clinical autopsy study. Three features in the clinical case. One is the endothelial cell dysfunction. Second one is smooth muscle cell, smooth muscle proliferation, and muscularization. The third one is the perivascular fibrosis. In your model, both the TWIST1 and the FOG2, do you have any data? These two pathways regulate the endothelial cell dysfunction?
We haven't looked specifically in those models to answer that question, but I think your question raises really interesting things about what do we know about the pathophysiology of CMVD, and some of these are from human autopsy samples and some are from mouse models, like a hypertensive diabetic rat, for example. And there's clearly evidence of smooth muscle cell proliferation, and that's why you get some of the luminal obstruction and some of the tortuosities that form. But I think we don't know. And when you mentioned endothelial dysfunction, on the autopsy samples, that's hard to assess unless you're doing immunohistochemistry and finding markers that suggest that. But one of the things the field is missing is the link between these structural abnormalities and the functional problems that we see and we measure and we evaluate in patients that we think are giving them the syndrome. So I think there's still a pretty big disconnect between those, and that disconnect also exists in coronary artery disease. And I think we've been able to bridge it a little bit better in CAD than in CMVD.
Second question in regard to the upstream, plaque and the downstream coronary artery vascular disease. Based on the clinical result, there is no correlation. Can you comment on that?
Sorry, there's no correlation between the presence of CAD?
Upstream large coronary artery plaque and
Yeah.
the downstream
Yeah.
microvascular disease.
Yeah, I think they're two different processes. They can coexist and overlap, but they don't necessarily have to. And I think about it a little bit like aortic stenosis and aortic sclerosis. So these are endothelial cells that line some sort of smooth muscle cell or valve interstitial cell group of cells. And they come from different embryologic origins. They're exposed to different mechanical forces. And that's why we end up seeing pretty different manifestations of the person's underlying hypertension, diabetes, hypercholesterolemia. So some people will develop really bad aortic stenosis and really bad coronary artery disease. Some will have one or the other. Same thing with CMVD. Some people will show up with only CMVD and the rest of their coronary tree looks pristine. Their valves are completely pristine. But I think they're just three different disease processes that affect three different regions because there are different developmental origins and they have different forces that affect them. So certainly they can coexist, but we see that they can also exist in isolation in patients.
Thanks, that was a great talk. I was interested to understand, recognizing that microvascular disease is sorely underdiagnosed, and it's been estimated that about 30% of women with ischemic syndromes go undiagnosed and may have microvascular disease. I mean, there's this search to identify women that we should be doing more advanced testing in and who may have microvascular disease. In your GWAS work, are you seeing the clustering of clinical factors like obesity, hypertension in that group with the mutation? Is that specific to African American women, or should we be, kind of thinking more broadly?
I think the GWAS studies don't specifically address that. I think it's a really interesting intersection of how risk factors are affecting the different genders, and certainly as you bring in other risk factors like obesity, how that plays out. But I do think that as we start to do more routine clinical imaging where we assess the microvasculature, like cardiac PET, or as we do more TIMI frame counts for clinical caths, which nobody does, but in theory we could all do, I think we'll start to understand more about what you're saying. There have been some studies that have looked specifically at your question, and I think the results differ. So in some studies, there isn't a huge difference between men and women, and other studies have actually found that there's an increased prevalence of risk factors in women, and these women are coming in and having decreased MVFR by cardiac PET.
Just finally, I wonder if you could help us with prognostic imaging. It used to be the dogma generated by people here 25 years ago that if you had a normal SPECT stress study that put one in a prognostically favorable group, and you would tell a patients that. I'm not so sure that makes perfect sense now based on the whole correlation between microvascular disease and less good outcomes, let's say. So is there a test? Is it at dynamic microvascular PET imaging? Is there a test that we can order and say, this is a favorable result, we think you're prognostically in a good group?
I think cardiac PET's a really good option for that. So the sensitivity and specificity for evaluating those perfusion defects is much better than SPECT. So you're not likely to say for a woman, oh, this is just breast artifact, I'll ignore it. The quality of the perfusion imaging is just better. But additionally, you get microvascular data. You get myocardial blood flow and blood flow reserve. And I think that information, as we collect more and more of it, will be able to answer your question much better.
All right, one last question.
One last question. Have to be jet here. No more. Actually, there were two very important questions raised here by Eric and Erica. And I think that's actually would be a good future direction for you. And that is that you have a high prevalence in African Americans. You can actually look to see if this variant actually differentiates between genders, number one. Number two, associated with obesity, metabolic syndrome, you can do GWAS actually on those.
But in our match
no
cohorts,
here, but you can go back to the UK biobank and use that data. You have only 44 here. The question I have though, among these all 42 that they were not having a congenital heart defect, how many were men and how many females? And your mouse models, did female have phenotype this small cell as well or not?
I think the match cohorts were about 60% female, and that's interesting in and of itself because the population is about 50-50 in the medicine biobank. So why it's 60% female? Interesting question. The FOG2 studies were not done in female mice. So it's a great question of what is the role of FOG2 in maintaining the coronary microvasculature, specifically in mice. And we know now that more and more journals are requiring us to look at these phenotypes in both genders, that there's huge variability in the manifestation of these transgenic phenotypes in the two genders.
All right, thank you.